And so we finally get to a structure that we believe is the fully formed structure. And this is a calculation. But what we see in this is secondary structure is forming. But through that long process, it took a while for everything to come together and come to the final structure. All right. But as secondary structure actually happens, we can see that the other secondary structure can make interactions with them. All right. So what really happens, okay, to get to the final conformational fold, all right, small areas of secondary structure can interact and can start to, once it's formed, start to stabilize and hold certain folds together while the rest of it can go through all the different combinations that it needs to find the full fold. And so there is a folding motif or a folding kernel that can form that is very stable as it helps progress the rest of it going. And so the kernels can actually, those motifs can be classified and will eventually get to the fully folded protein. Now, this is a really complicated process. Now, the native form is traditionally the most energetically stable form. The most unstable form is when it's totally opened to solvent. All of the hydrophobics are making interactions with the water. Those are very bad interactions, but if it folds completely, it is the most stable fold. It can be represented by an energy graph, and you can see way down at the bottom is the native structure. The animation will show you this can be modeled in 3 dimensions. And you can see as it's going through, if it's totally open, the energy is really bad. But as it folds together and you get some secondary folding, it can get to its maximal energy stabilization. Now, here's a couple things. Places like this or places like that, parts of the molecule, parts of the protein become very stable, but not all of it. Also, it can be represented here or here. These areas can be considered kinetic traps. Okay? Where part of the molecule is really stable, the rest of it isn't. But that stability of that small area, okay, you actually have to input energy to break apart that really good interaction that is incorrect so that you can make a better interaction later. Okay? And so some proteins, and what happens is the larger the protein is, the more kinetic traps can be formed that gets portions of the molecule in really stable conformations that are not the right full conformation. Now, when we did the experiment with ribonuclease A, it was a relatively small protein. And we confirmed that the amino acid sequence alone is the only information there. We can unfold it using chemical means, and then we can remove the things that unfolded it, and it will fold right back. There was nothing else in the test tube to cause the correct fold to come back other than the amino acids interactions among themselves. And it became fully functional. Fortunately, it worked, led us down this path. It only worked because it was very small. The larger you get, and the types of proteins that we have in a mammalian system are very complex. The larger you get, the more kinetic traps you can get. And if you unfold it, it may not be able to fold correctly by itself. The information for its full fold is in the amino acid sequence, okay? As long as all of it gets to that place. So in more complex systems, we're going to need helpers. And we'll talk about the helpers that would help a protein get over a kinetic trap and try to do something better. Okay? Now, in that collapsing portion, as the protein comes together and forms a globular protein, it gets closer together. The first driving force is the hydrophobic collapse, or putting the hydrophobics in the middle, trying to get them away from water. What happens is, as you start getting secondary structure, there's a lot of movement. If you looked at all those animations, it's still wiggling around. Okay? When you have that hydrophobic collapse and you have that wiggling around, what you create is a term called a molten globule. It is a protein that has collapsed, has secondary structure being formed, but is not the full fold. Okay, it can be about the size of what the protein should be. I know I have secondary structure, but a molten globule is molten in that hydrophobics are moving around. Those are not really locked in, and the protein will not function because protein's function is absolutely dependent upon the three-dimensional structure. And if it's got a moving three-dimensional structure, it has no overall function. Now we can study this by adding conditions to denature or unfold these proteins. And so a couple of the things that we've used very easily is temperature. And if you heat a protein up, you will unfold it. It will become denatured and it will lose its activity. So a lot of times when you are working with proteins, we do it on ice. Okay? Or you put it in the refrigerator. Or if you're in grad school, middle of summer, I'm walking into my department with a parka. Okay, because we would go into a room which is basically a refrigerator, and I would spend all day because I was isolating a protein and we kept it at refrigerator temperatures so that the temperature wouldn't destroy the protein as I was isolating it. Okay? Just kind of oddball. Everybody's in shorts and I've got a parka going in. All right? So we try to keep temperature as part of the process to keep the structure good. Okay? Cooking. Really, cooking, one of the purposes of cooking is to make the food safe. And to make food safe, one of the processes is to denature all the proteins that are there, including viruses and bacteria and things that aren't there. We cook them until they are purified, quote unquote, meaning that we destroy the activity of the things that are there. All right, another thing that we've used, urea. That was in the experiment with the ribonuclease. Urea disrupts the hydrogen bonds that hold proteins together. That will unfold it. Another thing here, guanine hydrochloride, that's what's over below, is a salt that will also disrupt polar interactions or hydrogen bonds also. And so any of the polar interactions, guanine hydrochloride gets in the way, and the more I add of that solution, the more I'll get a protein unfolded. And we can get these curves. A curve that shows the denaturing. And we can actually, for each different type of unfolding, we can get a melting temperature, which is what we use with DNA, okay, or a unfolding concentration that will represent 50% integrity. Now, the molten globule can be identified. Now, this is an experiment called circular dichroism. Okay, so circular dichroism. All right, what we do is we have a long tube. Okay, and on this tube we have light coming in. But it is very special light, it's polarized. Okay, and then on this side we have a detector. And the sample's inside the tube. Now the polarized light means that all of the wavelengths are in one orientation. Okay, and so we can measure that with a detector, know what orientation it is. I can send that light through a sample, and if by chance the light hits something that is optically active, the light can be twisted, okay, and come out the tube on the other end. And the amount of twisting can represent how much is in that solution that is, let's say, handed or chiral. All right. You use this in organic chemistry to look at optically active or chiral molecules, and that instrument was called a polarimeter. Okay, and that polarimeter could tell you how much R and S were there, or if it's a racemic mixture, there are chiral molecules, but it gets bent one direction, then it gets back the other direction, and then it ends up coming out straight. Or if it doesn't bend at all, then it may not have been chiral at all. The same thing here. Now, the most or the largest thing that will cause the light to twist is the alpha helix. They're all in a twist, all in the same direction. Okay. So what happens is, in circular dichroism, there is an absorption. Okay? An absorption that has two maximums, here and here, that will demonstrate alpha helix. In the native form, if the protein has alpha helix, the native form you would assume has the greatest amount of alpha helical content. Okay, so the native form fully folded. Under guanine hydrochloride conditions, we don't see that double dip, and so we have no alpha helix because it's been totally unfolded. Then there are times in which there's a condition that creates secondary structure, but it is totally inactive. Okay, and so what we're showing in this, under acidic conditions, a different condition, this is the molten globule state. Okay, no tertiary structure, no secondary. Has secondary structure because I see alpha helic content, but it has no tertiary structure. Native has both secondary and tertiary because I know it functions. All right. This example here is not what you would typically expect. Okay? If you had to guess, a partially folded protein should probably have an absorption like this, where you have fully native, lots of alpha helix. Partially folded, part alpha helix. Unfolded, no alpha helix. Okay? In this case, in this example, the molten globule state actually has a little bit more alpha helic content. All right, how could that happen? It's not what you would expect, but it could happen. All right, I have the collapse hydrophobic. I start making alpha helix. One thing about alpha helix, remember all of those twists have all of those hydrogen bonds? Alpha helix tends to have a lot of stability. And so once you start making an alpha helix, amino acids like to stay in alpha helix. And so if you've collapsed it and you're starting to form alpha helix, there is the possibility that you make an alpha helix and it goes a little bit more on this side and a little bit longer on that side, okay, because it's loose, it's open. For it to be fully formed, it has to collapse a little bit more. What happens, some of those alpha helix have to break or stop earlier so that the full form can take in its more compact position. And so you could, by chance, have alpha helix because they tend to like to make. Okay? That is not what you would expect, but this experiment shows this. Okay? The molten globule is a state that is between the random coil and the native state. For most proteins, the molten globule state lasts faster than we can see it. Most proteins fold very, very quickly under normal conditions. Okay? But it is a state where I'm in a partial fold. And so there are researchers that try to identify proteins that can be stabilized in a molten globule state so that we can study it. So we can study how folding actually occurs. Okay, how's it formed? First, the hydrophobic collapse. Then we have secondary structure formation. Alpha helix, beta strands, beta turns are likely to form. And then the tertiary start to make interactions. We start creating what's called super secondary structure, and in older terminology, we might call them folding motifs, or now we call them domains that tend to fold and create information. There are a lot of domains that have been identified that reoccur between proteins. One of which is a 4 helical bundle, very, very stable, seen in lots and lots of different proteins, and they create a very stable core. So here's one that we can see. Beta strands tend to come together, and they create a Greek key. And so in this, the Greek key, you can see a motif that is seen in Greek architecture. What is actually happening in B are 4 beta sheets coming together in a similar pattern, and then the A is what it looks like in its fold, but the Greek key is something that's seen quite often and folds very quickly. There is all sorts of different folds that can happen, and you can see how you have beta sheet, alpha helix, beta sheet, alpha helix in a repeating pattern. It can get longer and create folding structures that are a little bit different, where the beta sheet in the side surrounded by alpha helix. There's all sorts of very complicated, and they start to get really complicated, domains that can be identified. And what this actually gives is it confirms properties to the protein. And so in this particular fold, it allows the protein to associate to membranes. That particular fold tends to bind to proteins on the membrane. And so if your protein has to be associated to the membrane, if you look at its structure, whatever it does, usually has this fold somewhere in there. It's kind of like the Lego piece that plops into that one holding spot, and it directs, or it's the address of where that protein needs to be. What we do quite often is we start to look at the sequences of the proteins. And just looking at the primary sequences, we can identify these domains. And so we can see all of these proteins, okay, that have lots of different names and actually lots of different processes, but they have one commonality. They're all bound to the membrane. And there near the N-terminus, I can see a sequential sequence, even in the primary sequence, of what that domain would be. And so if I find a brand new protein that I don't know what it is, but I see that particular sequence, I might assume that's associated with membrane proteins. So then I might start looking at, well, what kind of membrane proteins might it interact with? And it helps us identify why or what that protein might do. Okay. There are other experiments that we will look at to try to identify folding, and so try to understand this. Trypsin inhibitor, it is a small protein, and here is the structure. You can see it's got 58 amino acids in it, so it's really a tiny protein. So starting at 1 and ending at 58. Now, for some reason, they identify lysine 15. I have no idea why they identify lysine 15. That's not important, but that's the picture they chose. What's important here are the disulfide bonds, because they are particularly stable. And so we have 1438, we have 5130, and we have 5 and 55. Okay? Now, 2 of these are embedded close to secondary structure. And one of them is not in a reoccurring secondary structure that we see a lot. Okay. And so, what the experimenters did was they tried to trap folding intermediates over time. Okay? So what they did is they had the protein and they allowed it to fold. What they're going to do is they're going to take, at time periods, a sample out of the folding test tube, and it's all trying to fold. And it's going to identify to see where the folding got to. Okay? The experiment that they're going to use here is they're going to separate them by size using a technique called HPLC, high pressure liquid chromatography. Okay? That's not important at this moment in time, it's just an instrument that will separate them by size. Here's the deal. Normally, a totally unfolded protein, if it tumbles in space, takes up a huge sphere. Okay, so it looks big, okay, even though it's going to weigh exactly the same as a fully folded protein that is really tiny, but when it tumbles in space, it makes a very small sphere. So it looks small. Okay, so we're separating by size, although they all weigh the same. It's how much volume they take up when they tumble in space. That's what we're really looking at. All right. What we tend to have is, as it becomes more fully formed, more of the disulfides are linked, because that stabilizes that whole structure. So what we're going to look at is see over time how many of these disulfide bonds are formed and which ones form first. Okay, that's what they're going to attempt to do. Okay, and they're not going to do it with one analysis. They're going to do it with many analysis over time. All right. 3 disulfides. Okay, when we reduce it or when we unfold it, it will lose its activity. Only when you have all 3 bound correctly will it be folded and functional. Okay, don't freak out. Here's the experimental data. All right, check this out. I have 1, 2, 3, 4, 5 time periods that I studied this, okay? So that's, every trace is a certain time period. Okay, so what do these peaks represent? All right, what I'm representing is separating it over size. Small comes out first, large comes out last. So the largest or the last peak represents the fully reduced or fully unfolded. The first peak represents in the native or the fully folded because it's the smallest. Then I got all sorts of intermediates in the middle. Okay. Now they did, after analysis, identify what these peaks represented. No disulfides. This peak right here, only the 3051. That little peak right there is the 55-5 only. This one that's represented right here, that's that second one and all of those, that represents 5 and 55 and 1438. This peak represents the 3051 and the 1438, and then this is all 3 of them. Okay? 100% of the sample is distributed between all of these peaks. Okay, so in this first run, the majority is still totally unfolded at 1 minute. I have some represented a certain fraction, that's only 3051. I then have some small fractions that represent 2 of them, and I have none that's fully folded at 1 minute. Okay, and so as the time goes through, by 25 minutes, I have absolutely no unfolded, totally unfolded protein. They've at least made 1 disulfide bond. Okay, and in essence, if you take a look, these represent 1s. I've made at least 2 by at least 25 minutes. Okay, but if I go back to 2 minutes, I still have a significant amount that's totally unfolded by no disulfide bonds. I still have some singles. My doubles are growing, and I start to see fully folded. And so what happens over time, this goes down. This tends to stay pretty constant until I get to most of it moving over. These will grow in intensity and become kind of constant for a little while, but they start going down as I start folding fully, and eventually I would expect that I would get complete native over time. Okay? What does this experiment say to me? And what does this experiment say to you? And does it say the same thing? Okay. All right, so you understand where the graph is. What would you have expected, or which disulfide would you have expected the first one to form of the three?
1438.
1438? Why do you think that one's the first one to form?
I feel like that one would have to form before to fold the rest of the protein.
Ah, to fold the rest of the protein, yeah.
3051 is connected to the alpha helix.
3051 has alpha helix and a beta sheet. The 555 is between two alpha helix. Now remember, what happens in protein folding, what we think. Hydrophobic collapse, secondary structure forms, the secondary structure kind of interacts by itself, it's moving around, and then everything gets locked down. The experimenters thought that the ones near the secondary structure would probably form first. If you can see this, the experiment that shows one linkage only was either 5 or 55, or 3051, both of them around the secondary structure. What we don't find is the 3814. Okay, that's in that random coil portion where it has a lot more movement. 1438 is only found when it folds around, right? And that's the whole deal. But it only folds around because these secondary structures are starting to make interactions. And when that secondary structure makes the interaction, it starts to make the 1438 come together more closely, and that can be locked down. And so we get one of the secondary structure disulfides around the secondary structure first, which then causes the 1438 to happen, which then finally allows the secondary structure one to fold. And so this tends to support that the secondary structure will be close, or that's the ones that get close together first. Okay? And then that, once you've got that one trapped, the 1438, there's a whole lot less movement than when it's like this. Once it traps over, that 1438 back here is much more closely aligned. Okay? And so, over time, just looking at the percentage of where it was, tends to support the idea that the secondary structures come together first in a folding pattern. Okay? Now, in the textbook, they talk about 3 models for which folding occurs. And they break it down and says, there's a group that says hydrophobic collapse occurs and then fold. And then they say another set of people say that the framework model, that you start to make secondary structure and that secondary structure will then start to assemble the tertiary structure. And then the nucleation model where it says, certain secondary structures form, they nucleate, or they create these folding kernels and they all come together. And I make the protein folding. Really, when people make a model, that's all well and good. Very often it is not one single pathway, but it's a combination of these. Certain proteins may rely a little bit more on one of these things in their folding pattern than others, but it's all a combination. Every protein collapses upon itself to hide the hydrophobics. Okay? As you bring things close together, the secondary structure really is optimized to form. Okay? And when that secondary structure forms, secondary structures are kind of close together they have the interactions that need to lock them together so you can start to nucleate or create the folding kernels. And so really, it is not, I wouldn't argue it is 3 separate models, although you might be able to find groups that will support 3 models. It is a combination of all of these things working together. And for most proteins, it happens on time scales that we can't even track it. It happens so fast. Now, as I get larger and larger proteins, some of those folding kernels can create kinetic traps, like we said. So we need some proteins to help us get past really good interactions that are the wrong ones, so that I can make more good interactions than just that single one. Here are some proteins. The protein disulfide isomerase, usually called PDI. The protein prolyl isomerase, usually called PPI, and molecular chaperones. There are a couple classes. The PDI operates this way. It has a sequence in its amino acid near what would be considered the functional site with two cysteines. They're highly reactive in that their pKa is about 7.3 versus 8.5. So if you go back to Henderson-Hasselbalch's equation, in normal solutions, about half of it will be deprotonated and about half of it will be protonated. Okay? And so I have a sulfur that is negatively charged that loves to react with other sulfurs. Okay? And so what happens here is I've got a protein that is bound with disulfide bonds, but it's the wrong one. Okay? And so there is strain in the system. Although once you get a disulfide bond link, it's a covalent bond, it's just not going to let go. Okay? But remember, this protein is under strain, kind of like this, like really under strain. It's linked, but it's under strain. So this protein will come in and it will break randomly disulfide bonds. And so it attacks, breaks the disulfide bond, creating one of the sulfurs internal to be negatively charged. And so I break and, okay, that's better, right? And one's attached to this enzyme. Now this one's free. And it says, oh, check this out. There's a sulfur right here. Let's try that. And it will make what would in essence be the right bond. Okay, much more stable, less strain, and that would move that second disulfide over so that it can react with the sulfur that it's supposed to be and release the chaperone or the isomerase. Okay, and now I've shuffled. Now what happens if this thing comes in and reacts again? What if it comes in and it reacts with this? I would get, oh man, don't do that. Okay. If it came in, I would get this form. All right. And remember, that was a good bond. Well, the sulfur is open, but it's in a good conformation. So it's not going to go up and create a bad interaction and make a bond up. It's going to go back and react right back where it was and kick that out. And so this isomerase is non-discriminating. It's going to go in and break a bond. If it's under strain, the protein that gets broken under strain, it will move. And it will form a different conformation. But if it breaks a bond that is totally good, then it will reform the bond it was just broken and it will go off. It's catalytic. It just goes around and breaks. Over time, it'll allow the shuffling of disulfides to get to the most energetically favorable. And it doesn't matter that you're breaking them, I'll get them all to the right fold. Okay. It can increase the disulfide shuffling by about 6,000 fold and lead to good folds. Okay. That's not the only way a protein can be unfolded incorrectly. All right. The protein prolyl isomerase, okay, works on prolines. Okay? Most amino acids are in the trans state. The exception is proline. 6% of all prolines in proteins are in the cis state. Oh, good night. Okay, do you remember what cis and trans is? Okay, same side, opposite sides. The same side of what? The opposite side of what? A what? What is that? Trans, right? And this is cis. Okay? But to have cis and trans, you have to have a what? Double bond. Does proline have a double bond? No. So how can you have a cis and how can you have a trans with no double bond? All right. Discuss with your partner. How can proline have a cis and a trans with no double bond? I'm just making it up.
This is the ring.
Oh, I also show a cis and a trans here. Try to pick out which one's cis. Try to pick out which one's trans. You got a 50-50 shot, right?
This one?
Uh-oh. There was some insight. I heard it right over here. Anybody ready to take a guess at it? All right, let's do this. Let's do the easy thing first. We can totally guess, right? Let's do choice A or choice beta. All right. Who thinks the trans is A? Okay. Who thinks the trans is beta? All right. The trans is A. And the cis is beta. Okay, so how? Why? That is the question. Yeah, wonderful question. All right, I'm going to show you where double bond is. There's a double bond. There's a double bond. The carbonyl is a double bond, right? All right. That doesn't help you, though, does it? No. Okay. What did we know about a peptide bond, though? Tell me something about a peptide bond. Between the nitrogen, the carbon of the carbonyl, and the oxygen, what is happening? Oh, yeah, what? Isn't there a resonance? And so the bond right here has a double bond character because it is resonating with the carbonyl. So much so that, remember, it doesn't twist around there. There is no angle, okay? And so in this case, if I have a big R group going this way and a big R group going this way, and this is the double bond, that's trans. And if I have a big R group going this way and a big R group, which is the whole protein, that is cis. And cis is, at least sterically, not as good as a trans. But the proline having a cis can overcome that bad interaction because a protein needs to fold and compact. And so all of the interactions that occur past that must be really, really good to overcome the steric bad interactions that would have been better in the trans. The trans, there's not a lot of strain around that bond, but there may not be lots of good interactions that occur, as well as when you compare it to this cis, all of these interactions might be very stable, and it overcomes. All right, so 6% here, 94% there. So 94% of the time, trans is better.